Hello everyone! Today we'll be reacting on videos featuring common laboratory tools and equipment in cell and molecular biology. We'll be reacting particularly on cell well plate, automated cell counter, transfer tank, ultrafiltration concentrator, and cryogenic storage. So first we'll be reacting on the cell well plate.
First, transfer 25 to 200 microliters of PBMCs into a round-bottom, non-tissue culture-treated 96-well plate. The cell suspension concentration should be 5 × 10⁷ cells per mil. Antibody cocktail to the cell suspensions and mix well. Incubate for 10 minutes at room temperature. Vortex the easy-set magnetic particles. Add the particles to the cell suspensions and incubate at room temperature for another 5 minutes. Bring the total volume in each well up to 250 microliters by adding easy-set buffer and pipette gently to mix the cells. Place the 96-well plate securely onto the easy plate magnet for 10 minutes. The magnetically labeled The newly isolated B cells are now ready in a convenient format for immediate downstream analysis.
Fresh ang mga gagmay nga test tubes.
Ako maingon kay mas easier siya sa katalantaw nato previously na dapat gamiton nato for this video. Kay naay magnetic thingy para makontrol ang number sa cells evenly. Pero mas kapoy siya kay tangtangon lang gyapon nimo ang excess na cells. Ito ba?
Sa ganito sa previous, supposedly atong katong gamiton nato sa previous no kay murag more on like mga do's and don'ts man to sa kung unsay.
Ang sa previous kay manual na pag cell seeding. So murag mano-mano pag pipette kay murag pipette up and down jud niya para ma-mix ang cells. Dali man gud mo lugdang ang cells. So kato, okay. Pipette up and down or even ang number.
Nakay maingon no.
Kato siya nga first nga video is kaning, di ba, murag double work na. Di ba, gipipette niya sa tagsa and then gipipette na pud niya multi pipette. Multi-channel pipette. Para ma-mix thoroughly ang kaning cell. So, kuan siya, double work na. Redundant na kayo. So, mas tsada yun itong multi-pipette ra diretso gamiton.
Kaning new. Diba?
Yeah.
So the next tool would be the automated cell counter. So, automated cell counter is a machine automatically count cells. So the sample is loaded into its automated cell counter and then it forced through a small tube. And then while the automated cell counter also uses optical or electrical impedance sensors to count cells. And then so the advantage pud sa cell counter is that it is 10 times faster. So 10 times faster than manual counting, and it can also count a large number of cells to increase the statistical accuracy sa study.
Nice.
Introducing the Countess 2 FL Automated Cell Counter. It gives you the flexibility to fit your cell counting needs and also your lab budget. Whether determining live and clear delineations of cell boundaries, providing accurate results even in samples with clumpy cells, all with eliminating the need for tedious manual counting. The Countess 2 FL Automated Cell Counter is small and easy to use. Simply turn on the switch and you are ready to start counting. For traditional bright field cell counting, add trypan blue stained cells to the disposable slides can also be used for added convenience and easy cleanup. To increase precision in the cell count, the Countess 2 FL counter automatically scans through the focal range of the sample to find the optimal focal plane. The Countess 2 FL cell counter provides an image of the results for visualization, showing the concentration and percentages of both live and dead cells within the population. The interactive screen allows zooming in on the image to get a closer look at the cells being. Countess 2 FL automated cell counter can be configured to perform a variety of fluorescence applications, including fluorescent protein expression, cell viability, or apoptosis. Simply open up the back of the instrument, add up to 2 light cubes, and the Countess 2 FL is ready to count fluorescent samples. Light cubes are available to detect a full range of fluorescence depending on experimental applications. The Countess 2 FL cell counter allows the simultaneous counting of cells using bright field and up to 2 fluorescent channels. Brightness of the fluorescent channels can be adjusted using sliders to increase or decrease the threshold for what is counted. Images of the cells with the data can be saved as image files, and numerical data can be saved as a CSV file on a USB drive for further analysis.
Okay ko.
Yeah.
Right, pero murag mahal kay siya tanahon. Kay grabe siya ka
Lagi.
high-tech. Ayos kay itong, wala ko nag-expect na itong pwede nimo siya ma-download diretso via USB, flash drive, something.
Ayos ko ang kanang parang daghan na fluorescence pipe.
Katong light cubes, no?
Let's say-
Itong 15 ka po.
Depende man gawin na sa stain, no?
Murag mo depende
Diba
sa imong
po?
sample kung unsa imo ang i-detect nga nga cells.
Oh.
Yes, and then kuan pud siya aside from daghan siya og features pa na. Kuan pud siya. Mas dali gid siya. Mas dali siya asa kuan compare sa manual nga kuan kay di ba? 10 times daw siya faster than manual counting.
Oh.
And then dayon di ba gi-cite pud dito giingon pud dito nga it can also count clustered cells like di ba maglisod man jud ta og count sa mga cluster cells nagkumpul-kumpul. So sa iya mas okay, mas paspas, mas dali.
Yes.
No.
Very
Technology.
useful.
Mahal siguro siya.
Very
Mahal
useful
dito
but
siya.
expensive.
Yeah. Okay, so for the next equipment, we will be watching the transfer tank. The transfer tank is actually used in Western blotting, which is a technique in molecular biology that is used to detect specific proteins in a sample of tissue or extract. So let's watch this video to learn more about the Western blotting technique and see this transfer tank.
As with all PVDF membranes, the GE high bond P blotting membrane must be pre-wetted in methanol, rinsed in distilled water for 5 minutes, then equilibrated in transfer buffer for 10 minutes. Always use a blotting membrane of the same size and shape as the gel and the correct size for the frame. Place the sandwich in the transfer tank. To avoid errors, always orientate the sandwich with the anode towards you. Transfer the proteins to the membrane with the desired voltage. Confirm the transfer by looking at transferred rainbow markers on the membrane. If it looks like this, you are doing well. Next, block the free sites on the membrane using ECL prime blocking reagent, which is compatible with your other reagents. Add the blocking agent and place on a rocking table. Blocking normally takes 1 hour at room temperature.
Sige, explain.
Actually, ang transfer tank kay kuan na siya, like sa wet western blotting siya na technique pwede. Kay naman gud na itulo ka western blotting technique na dry o na semi-dry. Na-mention to niya sa sugod. Ang wet lang at western blotting technique ang gagamit sa katong transfer. Ginabutangan siya og katong katong transfer buffer ang tank kay mo man siya ang ginagamit para mo balhin tong katong ibalhin ang iyahang proteins from gel to membrane. And ansa pa to? Ang sandwich kay kuan man gud siya di ba ang ibutang niya kay murag ang sandwich bali is like sponge and then the paper, then ang gel, then ang membrane, then paper na pod, then sponge. So bali giipit siya then ibutang siya dito sa transfer tank para pag-transfer from gel to membrane. Yes.
So ako pong maingon is, 'di ba, useful siya nga kuan tool since it separates and identify complex proteins. So meaning kuan kani maskin sa mixture ba daghan siya og marag complex ang mixture sa usa ka protein, maka able Japan siya to identify sa mga proteins, specific proteins.
Yeah.
Yeah.
Okay.
Yeah.
The second to the last tool would be the filtration concentrator.
In this video, you will see how to concentrate your protein solution using the Thermoscientific Pierce protein concentrator for sample volumes up to 20 mils. Do you want to desalt or buffer exchange your protein too? Not a problem. The Pierce protein concentrators will simultaneously concentrate your protein while removing salts, allowing for easy transfer to a new buffer. These disposable devices are conveniently used in a benchtop centrifuge, a fixed angle rotor that accommodates 50 mil conical tubes. Make sure that the concentrator has a proper counterbalance. Centrifuge up to 5,000 times G until your desired concentration factor is achieved. The dead stop volume is 50 microliters. The retentate volume can be visually monitored through a clear window with graduations as little as 15 minutes with protein solutions of 0.1 mg per mil or higher. That said, concentration times may vary significantly based on molecular weight cutoff and dilute the retentate to the original volume with exchange buffer and then cap and mix. Repeat centrifugation followed by addition of exchange buffer until desired solute removal has been achieved.
Para siya sa protein and salt.
Dili kuan siya. Ang salt bali kay imuha siyang pagkasabot nako kay murag itanggal nila ang salt.
Maka-filter siya of salt.
Muna
Salt.
katong iyang giingon nga 98% kay murag mas mas ma-remove ang salt concentration ata.
So,
Salt
sa isa ka
concentration.
solusyon na naay protein ug salt, tantangon ng protein ug ang salt. Sakto ba akong pagsabot?
Simultaneous. I don't know. Not sure.
Di po ko sure. Pero murag ang ako pagkabalo kay inatanggal nila ang salt, then ang murag ang ma-retain ang katong protein. Mo to katong retentate, katong mabilin. Ang target protein bale.
Okay. Okay.
I'm not sure. Pero mo
I'm not
sabot
sure.
sa video.
I'm not sure.
Okay, next na ba?
Okay. Mag-next na ta. So the last laboratory equipment we will see is the cryogenic storage. So I don't know much about this equipment except for the fact that, as the name suggests, it is used for storage. And since it's cryogenic, maybe it is for long-term storage use. So let's see through this video.
Is liquid nitrogen down. There's a temperature difference between the liquid nitrogen and the air is now. Now that I've cleared the vapour that was on the top, I can see my samples. Got little tubes that have a small amount of sperm in there. Something like this, which is called a dry shipper, and we can fill the dry shipper up with liquid nitrogen. The liquid nitrogen gets absorbed into polystyrene. These samples bung that can go on the top to keep the samples nice and cold. The liquid nitrogen that is in here, it keeps the samples cold enough for about 3 days transportation, which is more than enough time to get these samples all over the world.
So kuan gid siya for like
Congrats.
long-term storage, like what I said. Kay kuan siya like gamit sila liquid nitrogen, that's why it's called cryogenic.
Pero daghan kay sulod ang like tank. Kay
Storage tank.
marag 76 daw ka racks.
Dako siya nga
Then, ang
tank
isa-
actually.
Dagan unsa niyo pag-locate sa imong sample?
Mo to yang gibuhat, katong iyang gipa-release gamay, nagpa-release siya gamay og kuan para marag mabawas-bawasan ang liquid nitrogen sa sulod since taas ang marag temperature sa to yang giingon. So para mas
Pero
sample. Ang mura pud ko katong kuan katong dry shipper nila nga dako kay ang butanganan pero gamay kay ang sulod. Like mura to ilang i-ship. Pero na-amaze kay ko.
pwede isa lang ka sample ba?
Gani. Dili po siguro oy kay murag malungkot. Ana may maingon lain.
Ako maingon is, very useful yun siya for, storage kay. Para ma-preserve nato ang gusto nato nga mga, like for example, genes ba to?
Murag ko na to mga cells,
Tissue?
sperm cells, and kung ano to yung gigamit. Basta ka na, eukaryotic and bacterial cell storage siguro.
Okay. O, atong ma-store ato ang kuan kaning gusto nga cells for the future.
Yeah. Na-search na ko ang iyahang price. Murag maabot yun siya mga 1 million ang isa ka tank. Yeah.
My God.
In pesos?
In pesos siya. Ang kay ang sa USD kay kuan man kanang pila to $23,000, $24,000.
So, I think kuan siya lain-lain nga sizes.
Yeah. Kita po ko ana. Na siya lain sizes. I don't know kung tapila tong na size ang naa sa video. So that's all, guys. And I hope you learned something from our video presentation. Okay, see you.